My project is called A Comparative Analysis of the Remote Touch Mechanism in Birds by myself, Suzanne Spriggs, and Dr. Cost. So first off, what types of feeding in birds are there? There's visual, auditory, chemoreception or smell, tactile or direct touch, and remote touch, which is what we are focused on. What is remote touch? Remote touch is when an animal, in this case birds, can sense an object without touching it by detecting changes in pressure. This allows information like distance and size of the prey to be collected before coming into contact. You can think of it like sonar, but it is functionally different. Remote touch is mediated by an organ called the bill tip in birds. The bill tip organ has been found in 4 families of birds: Apterygidae or kiwi, as seen here, Salopicidae or sandpiper, as seen here, Anatidae or waterbirds, as seen here, and this is a mandarin duck, and Threscanornithidae or large wading birds, and this in an BC. All of these families use probing or poking to find their food, meaning visual cues are very limited, if any. The bill tip in all these species is filled with mechanoreceptors, specifically Grandry and Herpes corpuscles, that allow for the remote detection of prey. So what are Herpes and Grandry corpuscles, and what do they look like? Herpes corpuscles are pressure-sensitive mechanoreceptors that respond to low frequencies found in various locations of a bird body, and were first described in a paper in 1848. Herpes corpuscles are ovoid in shape with a cylindrical axon terminal, as seen to the photo to the right. This photo is from a transverse angle, meaning you are looking at it from the top down. Each axon is surrounded by a bisected lamellar inner core, or 1 in this photo, inner space, or 2 and 3 in this photo, and capsule, or 4 in this photo. The large outer lamella, or 3, contain incomplete lamella and a few organelles, while the smaller inner core, or 2, contains more distinct complete lamella. On the other hand, Grandry corpuscles, as seen here, are tactile mechanoreceptors found mostly in the distal subepidermal beak skin, or the skin closest to the tip of the bill. The first drawing of a Grandry corpuscle was published by Grandry himself. A Grandry corpuscle is in the photo shown to the right, which is also seen at a transverse angle. Grandry corpuscles are composed of a connective tissue capsule surrounding a layer of Schwann cells, or 3 in this photo. The actual Grandry cell is number 1, or the red ovals, and you can see that there are 2 inside this capsule. In a Grandry corpuscle, you can find 2 to 5 of these Grandry cells. Number 2 is pointing to a nerve ending between the 2 Grandry corpuscles, and there is always 1 left nerve ending than the number of specialized Grandry cells, so in this case, there's 1. Both Herpes and Grandry corpuscles are innervated by rapidly adapting afferents projecting from the trigeminal ganglia, meaning they send signals to the brain via the ascending trigeminal nerve pathway. Crocodiles as a comparison. If our study is on birds, why are we looking at past research on crocodiles as a comparison? That is because crocodiles are reptiles, and reptiles and birds are very closely related. Also because there is more complete information on crocodiles than birds. Crocodiles do not have Herpes and Grandry corpuscles, rather they have dome pressure receptors, or DPRs. DPRs on the cranium allow for the detection of surface pressure waves, allowing the crocodile to orientate towards the location of the water disturbance. Dome pressure receptors have a similar structure to Grandry and Herpes corpuscles, as seen in the photo to the right. This photo is from a sagittal section, meaning you are looking at it from the side, so it may be a little hard to see the resemblance. DPRs have a smooth dome structure with an apex, which is at the top, that allows deflections to be sent to the brain and are connected to a hypertrophoid nerve system, meaning in a large nerve system. Herpes and Grandry corpuscle location depends on feeding strategy. It has been shown that in the domestic goose, Grandry corpuscles are 10 times more abundant than Herpes corpuscles and are concentrated in the tips and ridges of the beak. However, in shorebirds, Herpes corpuscles were more abundant and are concentrated in the tip of the bill, and in grain-feeding songbirds, the mechanoreceptors are located only in the parts of the beak involved for seed opening. This photo is of the different mechanoreceptors in the duck and quail beak. The percentages are of relative area of the mechanoreceptor in comparison to the bill area observed. As you can see in the duckbill on the top, herpes and granary corpuscles are located throughout the bill, presumably in the ridges. In the quail, which is the one on the bottom, however, the herpes and granary corpuscles are located only in the tip and cranial regions. Is the abundance of herpes and granary corpuscles any different in the bill tip organ? This data is from a study by Burkhout. In a mallard duck, the bill tip organ is located in the maxillary nail of the mallard duck. A photo of this is shown to the right, and you are seeing the top of the bill. The maxillary nail has been circled, and as you can see, it is located at the end of the bill. In the bill tip, the amount of herpes and granary corpuscles is much greater than anywhere else on the bill. In the mallard, the bill tip organ is comprised of 45 derma papillae that are covered by keratin caps, as seen here. A photo of a single papilla is shown to the right. In the inside of each papilla, herpes and granary corpuscles are stacked around a central branch of afferent nerves, and there are 3 to 16 herpes corpuscles located proximally, and 4 to 18 granary corpuscles that are located distally, also shown in the photo. Crocodile mechanoreceptor abundance comparison. The photo below shows the density of DPRs in a crocodile obtained by Leitch and Cadiana. In this paper, DPRs were referred to as ISOs, which are the same thing as DPRs, just a more recent name. The scale to the right shows the density of DPRs, with red being the greatest density and dark green being the smallest density. As you can see, DPRs in crocodiles are highest in density around the teeth, and inside the mouth around the mandible and maxilla, which is similar to what we just saw in birds. How do herpes and granary corpuscles send their observations to the brain? The trigeminal nerve. The trigeminal nerve is cranial nerve 10. The herpes and granary corpuscles project to the brain by means of the trigeminal nerve. The trigeminal nerve sends information to the principal sensory trigeminal nucleus, also called the PRV, which relates to the end station called the vasa rostralis, also called the BAS. The BAS contains the somatomatic map of the bill tip. An example can be seen to the right. A somatomatic map is a region of the brain where each section corresponds to a different location, and based on the size, you can see the relative importance of signals from a given location. So in this example, we can see that signals from like the face and lips are a lot more important than, say, the signals coming from the foot and toes. Going from the brain out, the trigeminal nerve splits into the ophthalmic, maxillary, and mandibular divisions. How does the trigeminal nerve system look in a crocodile? The photo below shows the 3 divisions of the trigeminal nerve in a crocodile. The nerves in blue make up the ophthalmic branch, the nerves in green make up the maxillary branch, and the nerves in red make up the mandibular branch. And as you can see, it's also very, very highly branched. So why should we care? The photo to the left is a nerve map of a duck, and the photo to the right is a nerve map of a chicken. As you can see, both are very basic and lacking the many small divisions of the nerves as we saw in the crocodile. These experiments may not have needed to go down to all the little branches, but there are still few complete nerve maps of birds in literature. As we saw a few slides ago, crocodiles have a high abundance of mechanoreceptors, especially around their teeth and their mouth, as seen here. And as a reminder, the red is the high density. We also saw a few slides ago that the nerve map of a crocodile is highly branched. And if we take special note, we can see that there tends to be more branches in the regions where mechanoreceptor abundance was high. If you are having a hard time visualizing that, here are the 2 photos overlaid. As you can see, areas of high mechanoreceptor density in red are highly innervated. We know birds have high mechanoreceptor abundance, so that they should also have more branching nerves similar to these, where mechanoreceptor abundance is high. So what's next? Due to COVID causing ACRE to be online this summer, we weren't able to do any of the dissections that we had hoped to do, so the study became more of a literature review. In the fall semester, we will continue this project as an independent study by dissecting various birds and creating nerve maps for these birds to help fill in the gaps in knowledge. I hope that through the independent study, I am able to do enough more research on this topic to turn it into my honors thesis. It is possible for future studies to be done, even if not done by myself, to measure the mechanoreceptor density in these birds also, to match receptor density and the nerve maps as we did with the crocodile. And then for acknowledgements, I'd like to thank these people. And then we're excited, and photos. And thank you for listening to my presentation.